the next generation of leader in plant health. This is not just a scientific pursuit, it's the cornerstone for the sustenance of our global ecosystem. The Plant Health Think Tank is dedicated to building a network of students and young professionals with sheer passion for plant health related topics. Through advocacy, public research, online workshops, and capacity building efforts, this think tank is committed to increase the awareness and connecting aspiring individuals with seasoned professionals in the field. Our objectives include network building, advocacy and policy involvement, public outreach and education, capacity building, research and dialogue, policy analysis, which all are based on the plant health. In our two-day virtual gathering, we have privilege of hosting two experts, each bringing a wealth of knowledge in their respective field. Before we jump into the presentation, we'll having a take some more moment to appreciate the wealth of insight they are to share with us. Our format for today is quite straightforward. First, we'll hear the individual presentation session from each of our esteemed speakers. They will shed light on the respective area of expertise, offering us a glimpse into the fascinating world of plant pathology. Right after that, we'll be having our question and answer session, including audience response. It means after the presentation of each speaker, we'll be taking some more questions to be addressed from the speaker side. Now, without any further ado, let me introduce our first distinguished speaker, Mr. Ritesh Yadav. Ritesh Yadav is a Ph.D. candidate at the University of Manitoba and currently serves as a tenure assistant professor in the Department of Plant Pathology at AFU Chiton Rampur. His extensive research experience spans over 7 years and he is affiliated with Agriculture and Agri-Food Sciences, Agri-Food Canada as a research affiliate. Ritesh's expertise covers a wide range of areas from rapid detection and diagnosis of plant pathogens to integrated disease management, participatory plant breeding, molecular crop improvement, proposal development, and project management. Currently, his research focus evolved around the development of a robust genetic selection model employing a joint GWAS-GS approach to enhance fusarium head blight resistance and winter hardiness in winter drum. This collaborative effort involves the AAFC Land-Bridge Research and Development Center, Winter Wheat Preserve at the University of Manitoba, and AAFC Ottawa. Additionally, this is also engaging in the identification of KSP marker for FHP resistance and frost-tolerant rosy. So, without any further ado, I'd like to give the floor to Mr. Ritesh Yadav. Dear sir, the floor is yours.
Thank you, Upendra, and Plant Health Think Tank for having me in this seminar series. So I would like to welcome all of the attendees once again, and so I think I shall start, right? Am I good to go?
Yeah, you can go.
So let's begin my presentation. So just briefly talking about my journey so far. So right now, as Upendra said, I'm a PhD candidate and I'm currently working as assistant prof, but on a study leave. So about my journey, I belong to farms on the eastern plain of Nepal, particularly Saptari district, and I belong to a farming community. So I think in some ways that inspired me to pursue my career in plant science. So about my education background, or just about my education, I did my bachelor's in agriculture from Institute of Agriculture and Animal Science, Tribhuvan University, Nepal, and right now it's already transformed into Agriculture and Forestry University, even though there are some issues again. And I did my master's in plant pathology from Punjab Agriculture University. Then I started working as a program officer and budget manager at one of the leading research NGO that's also popularly known as Lever Nepal, or Local Initiatives for Biodiversity Research and Development. So, talking about my experience at Lever, I gained a lot of insights on participatory plant breeding from Dr. Bhuvan Ratna Stafit. He was a principal scientist at Biodiversity International, but he is no more with us. And I worked extensively on neglected and underutilized crop species such as foxtail millet, proso millet, and a lot of like beans, amaranths. And then again, I pursued my career in academia and ended up being at Agriculture and Forestry University. And right now I'm a PhD candidate, and I'm also working as a research affiliate for Agriculture and Agri-Food Canada. That's the lead agriculture research institute in Canada, just like NARC in Nepal or USDA in US. So just briefly talking about my thesis project. So it's on genomic selection for improving winter harness and fusarium head blight resistance in winter durum wheat. So just to give you a brief background about my project. So right now, Canada is the leading producer and leading exporter of durum wheat. It's also popularly known as pasta wheat. And right now, Canada doesn't have any winter durum cultivar. So this is the first kind of the project in Canada, and I'm really fortunate to be involved in this. So Canadian breeding program, it focuses on two priority traits for winter wheat, that's fusarium head blight resistance and winter harness. So these are the, so you can see some disease spike, right? So in this disease, there's premature bleaching of the spikelets, and this disease is economically important for yield losses as well as quality losses, because the fungus associated, that is fusarium graminarium, it produces a very harmful mycotoxin that's known as deoxynivalenol, that's also known as DON. So due to several reasons, it is very important in North America, particularly due to quality losses as well as yield losses. And similarly, winter hardness is one of the biggest priority traits. So I'm focusing on them. And as I said, right now there's no any winter durum cultivar. So this is a very, I would say, complicated topic, but I would try to simplify as much as possible. So we might be already aware of marker-assisted selection. So there are different kinds of genetic markers that are used to enhance the selection accuracy for, I would say, oligogenic traits that are governed by few genes. But fusarium head blight resistance and winter harness, they're genetically very complex because they're governed by many genetic loci, and they're affected by environment to a large extent. So that's why phenotyping is pretty challenging for both of the traits. We need a lot of resources and money to phenotype and to evaluate our materials. So genomic selection, it is I would say, simply selection based on the genotype or the DNA information of the breeding materials. So we might be already aware of some kind of regression equation. With the help of some regression equation, we can definitely predict some dependent variable if we have some independent variable. So in this case, so we create a training population that's also known as reference population. So generally, breeding programs they make thousands of crosses, and as a result of which we get large segregating population or breeding lines, maybe 50,000 or 60,000, and it's impossible to phenotype them, and because it needs a lot of resources and the phenotyping is really complex. So genomic selection is a kind of genomics assisted breeding technique or tool that aims to reduce the breeding cycle and ultimately gives us higher selection accuracy and ultimately higher genetic gain. So basically, suppose we have 50,000 lines. So we sample some representative lines from that 50,000 lines, population of 50,000, and we create a reference population. So in my case, the reference population is about 600 lines. So I phenotype them, or I screen them for FSB resistance and winter harness in the field, in the greenhouse. So I'm already done with my 2 years of phenotypic evaluation, and by the end of this October, I'll be having all of my data. So once I get all of my data, phenotypic data, so we have different genotyping platforms. Uh, you might be aware of something known as next generation sequencing. So basically, there are different kinds of sequencing technologies, such as Illumina or PacBio, or there are many more. So I already have the DNA information for my 600 lines. So once I get all of my phenotypic data, I implement different models. So those are really sophisticated models. Generally, they are, we call them, some of them, we call them parametric models, and some of them, we call them non-parametric, or generally we use machine learning algorithms. So with the help of that, we create some kind of prediction equation, right? And that equation gives the breeding value. So when we have that prediction equation on or model, so right now so as an instance, I gave you 50,000 population, right? So we don't need to phenotype 46,000 or rest of the, 44,000 or whatever is the equation after subtracting 50,000 lines, right? So we can get the breeding value for rest of the breeding lines with the help of that equation without phenotyping. So phenotyping, it takes a lot of time, a lot of resources, So that's how we can select the materials. And so since we reduce the breeding cycle, our selection accuracy is increased, we can select more accurately. So that leads us to higher genetic gain. And that's how we can feed the growing world population in the wake of climate change and different economic crises. So I won't be discussing all of them. So So the ultimate goal of my project is to develop winter durum wheat germplasm so that Canada, it will have some winter durum cultivars in place. So I won't be discussing the methodology, but I do a lot of I prepare a lot of inoculum for 4 isolates of Fusarium graminearum. In a season, I prepare like almost 10 to 15 liters of inoculum Fusarium. Then I phenotype them at 3 locations. It's pretty intensive and I would say cumbersome process. And then when the seeds are harvested, we analyze them for mycotoxin using ELISA, and that's really time taking. So that's why with traditional breeding or just phenotypic selection, genetic gain, they're very slow. So with the help of genomic selection, we can definitely enhance the genetic gain and can have some superior crop varieties in less time using less money. So as this webinar relates to plant health, so plant health is a very, I would say, big topic. So it encompasses several aspects of crop production, from agronomy to physiology to pathology to entomology, and so it's very hard for me to embrace everything in this short presentation. So FAO reported that so every year they estimate that there's crop losses of 20 to 40% due to disease and insect pests. And apart from them, we have furthermore challenges such as weeds and definitely climate change induced stressors such as drought or erratic rainfall or terminal heat stress. So they all are big challenges to global food production. So that's why it is very crucial to study these challenges or to consider these challenges and try to come up with a eco-friendly and economically viable solutions so that we can help the world feed, and at the same time we want our environment safe. So right now, just talking about a little bit of climatic adversities the world is going through right now. So Canadian prairies. So prairies, they are plains of Canada. So prairies includes 3 provinces in Canada. That's Alberta, Manitoba, where I'm right now, the coldest place in Canada, and Saskatchewan. So so these provinces or prairie, they are facing this problem of drought, and that's why. And that's where my project comes into play, because winter cereals, they have advantage of higher water use efficiency and less disease pressure and other several benefits. So that's why we are trying to create something that will be helping the prairie farmers. So as I said, durum is a big crop for Canada because it is the leading exporter. And similarly, in back home in Nepal and in Africa, so droughts, they are becoming pretty rampant. So that's why when we talk about plant health, we just don't stress on biotic stress, or we also consider abiotic stress, because ultimately both are going to cause heavy yield penalties or quality losses in different crops. So So there's this concept that's known as integrated plant health management toolbox, and we might be well aware of this disease tetrahedron, or sometimes we simply call it disease triangle. So there are three important components of any disease epidemics, or obviously insect pest epidemics. So we need all of these components: environment, host, plant, pest, in alignment with the conditions of disease developments. Generally, environment should be conducive for disease or pest epidemics. Host, they should be susceptible, and of course, the organism that's harmful, or plant pest, they should be virulent, or they should be able to cause some damage. So when all these conditions are fulfilled at right time, or with right frequency of those certain favorable environmental factors, we generally get disease epidemics or pest. And so we have different kinds of tools and methods to manage them, and generally these are very, I would say, these are very important to manage different kinds of stress, particularly biotic stress. So regulatory measures, it includes quarantine measures, and of course different kinds of regulations, such as phytosanitary measures and cultural practice. So definitely it's related to agronomy and soil science more. So different organisms, they are favored by different environmental conditions. So just by modifying our agronomic practices, we can actually avoid them, or we can reduce the risk of epidemics. And my favorite is genetic host resistance. So it is the most economical, sustainable, economically viable approach to manage any kind of biotic stress. And that's what I'm working on right now. And there's a lot of interest on biological control. So generally, so previously biological control was just about harnessing some organisms that's used to suppress the enemies of the plant. But right now, a new research area is emerging that's known as microbiome research, or from breeding perspective, there's a lot of interest on this microbiome assisted breeding. I'll be discussing about this a little bit in upcoming slides. And of course, disease forecasting models, early warning systems. There's a lot of things happening in this world. A lot of satellites are being launched. So we use different kinds of drones or satellites. We the people, they train different kinds of model, and that's how they can predict the disease or pests. And of course, along with them, they provide decision support system to the farmer so that they can act accordingly. So these are some of the emerging research areas in plant health management. And probably you might have you might have some idea about it. So microbiome research. So generally there's a lot of microorganisms inside and outside plant. So they actually help the plant in, some of them help the plant to grow better, or they also provide protection against different microorganisms or insect pests, and some might be harmful. So right now, researchers, they are working on identifying the plant traits that actually harbor, or that actually, I would say, attract beneficial microbes that help the plant with higher water use efficiency, or higher nutrient use efficiency, or plant health, or better crop resilience. So this is one of the area one might think of um, while considering graduate studies. And the next is predictive and genomics assisted breeding. So there are different tools in genomic assisted breeding. So GWAS, or you might call it GWAS. So it's on known as genome-wide association studies. So in simple words, it just like plant gene detectives. So they work like detectives. We use different um, mathematical models, and we try to pinpoint specific genomic reasons or genes that makes plant adaptive or more resilient. And similarly, genomic selection is other tool. So in my research, I use both of them, and with development in AI and gene editing, so things are becoming better with predictive breeding, but still we will need to wait for better, I would say, results and insights on those. So CRISPR-Cas9, it is being widely used to edit genes. So there's a big difference between transgenic and gene-edited crops. So in transgenics, we actually uh, introgress or incorporate some genes from foreign organism. But in case of gene editing, we just edit certain portion of the genomic regions or gene. So that's more or less equivalent to natural crossing that's happening in the nature itself. So right now, North America, it has relaxed provisions for genome edited crops, but still Europe, it's very hesitant to adopt genome edited crops. Similarly, nanoparticles, nano-based sensors, they're being used for plant health monitoring and, of course, management. And as you know, the world is now entirely into AI and machine learning. So, of course, these great tools, they help us to analyze large amount of data that's not possible with any statistical tools. So these are making a big difference in, I would say, all aspect of plant research as well as animal research or human research. So I think this slide should be particularly very interesting to the attendees. So for people who are seeking career in plant science or plant health or whatever, and intends to pursue a graduate program in some good universities. So these are the things one should start considering early. So generally, I would say GPA should not be issue for many people, but still try to maintain a good GPA. Generally, most of the universities, they have this eligibility of uh, GPA of 3 out of 4. And I think most of the students, they lack or they miss out on building relationships with profs and research scientists around them. So this is very important because you will need some profs or some referees who can write you a good recommendation letter when you are actually making an application, or when you're just emailing a prof. Sometimes they might uh, ask you a recommendation letter right away. And of course, I think now things are better in every part of the world, in different universities. So undergrad students, they get a lot of opportunities, particularly as an intern or summer school student, or just volunteer um, in different labs of profs or research scientists. So this is really important. So if you are involved in some research project, even if you don't get any stipend or any kind of incentive in terms of monetary value, but I highly recommend that you go for it, because it will help you to enrich your CV, and of course, you will gain a lot of research experience. And yeah, attending conferences, seminar, and webinars, they're particularly important because they, apart from helping you to get updated with current advances, these events also help you to network with different props, students, or other people. I would say connect with your seniors or alumni who are pursuing or who have already pursued their studies. And I highly recommend people to have some skill set in some programming or some statistical software, such as R, SAS. I would say SAS might not be possible for everyone, R and Python, because these days all of the research, they require some kind of programming or some kind of modeling. So this would be a really good value addition, and this makes a big difference when it comes to graduate school application. So so what are the things to be considered prior to corresponding profs or making graduate school applications? So I think one of the biggest problem student face is identifying their interests and goals. So to my experience, most of the students, they are pretty unsure about their interest and what they want to do in their career. So they might just say that a broad discipline, plant pathology, but there are hundred of research areas in plant pathology. You should at least have an idea of some of the topics, maybe post-pathogen interaction, maybe disease forecasting, or maybe biocontrol of plant disease. So you should have some specific research area in your mind before corresponding props. I don't recommend people emailing props randomly. So before preparing to email, study about props, research area, their publications, their lab facilities, and try to find a prof whose research actually aligns with their interest. Because even if someone gets lucky and get into graduate school, but if the research is not of interest to the student might feel confused in the later stage because I would say graduate program, it requires a lot of patience, endurance, and a lot of motivation. So generally, students who get lucky, as I said, uh, they actually struggle with motivation in later years, and that's really not good. And of course, before writing, if an email, if you have a language proficiency score at least, that is a great advantage. So that generally increases your chances, uh, to my knowledge. And when I talk to profs, so because profs, they want international students who can apply right away, and they don't want to wait for students forever. So if you have language proficiency, so generally in most of the North American universities, GRE is waived. So try to have language proficiency score in your hand before emailing. So to my knowledge, these are some platforms you can explore to find some openings. LinkedIn is a great way to get connected with profs. You can follow a lot of pages, especially pages of professional associations, societies. Twitter again, or right now we call it X. So try to have a Twitter account, follow the profs. So generally, profs they make tweets about their any openings in their lab, or of course they do its publications. So it will help you to get updated. Indeed is another I would say nice platform. And as I said, if you go through web pages of different professional associations or societies, like American Phytopathological Society or National Association of Plant Breeders, Canadian Society of Agronomy. There are a lot. So it depends on you in what you are interested. So you can go for it accordingly. So these are again some great websites. You can find a lot of openings in. This is arabidopsis.org, or you can just Google TAR jobs. You'll get a lot of openings for graduate research assistant or teaching assistant or other things. Brain Gene Jobs, it is again a good website. I would highly recommend that. And of course, if you are interested in a particular school or university, you can get a lot of information, or there are some link such as career or openings. So try to check them as well. So writing emails to props, it is really writing emails is a craft. So generally, people, they don't invest a lot of time writing emails because it doesn't hold true to everyone, but many, they just randomly email without giving a second thought. So this is very important. So as I said before, writing an email, if you're interested in a particular lab, try to read about the projects ongoing in that lab. Try to study the profile of the prof. What are the interests? What are the research area of the profs? And then only plan to email. So one of the common mistakes, or I would say a blunder people make while writing emails, is that they don't care for typos or grammatical mistakes. And if there are typos or grammatical mistakes, props, they generally don't like you, or they don't feel that people who are emailing them for graduate student, like for assistantship or funding, they're not serious enough. So these are, I would say, some outline of a typical email if someone intends to write to a prof for graduate assistantship inquiry. So please be clear about this subject line. Just write graduate assistantship inquiry. And of course, to my experience, if you write your research interest area, maybe phenomics, genomics, or biocontrol, or machine learning, so it depends on you. And of course, use a very proper salutation. So generally, students, they use dear props. I recommend not using dear props, because many props, they don't take that in a good way, you know. So just write hi. So many props might like it, but I would say many props they don't like, because dear in North America is used for someone who is very close, or it can be used by a friend or a family. So just use hi, doctor, the last name, or just use my supervisor's name is Kurt McCartney. So I write just hi Kurt or hi Dr. McCartney. I never use dear. And of course, while writing introduction, you have to introduce yourself in a very formal tone. Don't write in an informal tone. Mention your academic credentials, your area of interest, explain why you are interested in the props research, or how you fit into his lab or her lab. Highlight your any qualifications like that is relevant to the project. Just don't write everything. So suppose if somebody is looking for assistantship in social science, and that person is mentioning that I have experience in molecular biology, it doesn't make sense. So try to be careful and critical in your emails, because this is your this is your only chance to impress the prof or convince him or her. And apart from this, I highly recommend people to attach their CV, and of course, a very brief research statement. So you can elaborate why you are interested, how you fit into the program, how you can manage research projects. You can also write about your social skills. That is also really important, because profs, they generally don't like lab rats. They want someone who is cooperative and understanding. If you have any language score or GRE score, attach them. And if you don't hear back on time, you can always write a follow-up email or a reminder in a very polite tone. And of course, timing of email does matter. So if you're sending email from Asia, you have to take care, or you have to consider the working hours of the prof in mind. So generally there's 12 hours difference between North America and Asia. So in Canada, the working hour starts from 8:30. I think in US, Ram might enlighten you better. So it should be the same. I will get back to your questions if you have any. Okay, there's no any message. So generally, students they use a lot of AI and Chat GPT write emails, but remember, like props, they're way ahead of you. They generally use GPT zero that's accessible to them, and that can that can that can destroy your chances to get into that lab, because academic integrity or research integrity, AI written text, they are also now referred as plagiarism. So just don't rely on ChatGPT or any other AI tools to write emails. You can definitely take help, but write your emails on your own. And generally, AI texts, they sound pretty weird. They're very robotic when you read them. So I highly recommend just don't use ChatGPT. And of course, so I generally get a lot of emails for reference or for different reasons from students. And generally, from my experience, so their CV, most of the students, they try to make a very fancy CV. I highly recommend to keep your CV very simple and clear, you know. And so if somebody is using like very fancy CV, that generally doesn't actually make props happy, because most of the props, they were born in 60s or like in 60s to 80s. They just want to keep things simple. And of course, I would say simplicity is always a good thing to have, you know. So I just kept sample of my CV. So you just you can just create a CV with Word. You don't even need to use any CV generating tools. You just mention everything in a clear way, highlight your major achievements, try to tailor or summarize information. Don't write very lengthy words. Or always focus on your research skills. If you have any publications, or if you have any publications in draft, or you can also mention them. And it's always nice to proofread your CV. So because if you have any typo in your CV, again, that will give a very wrong impression about you to profs. And try to have references in your CV who actually are willing to offer the LORs or reference letters. Because sometimes what happens, like you might have some props in your CV while corresponding, and when you make the actual graduate school application and you change your references, so that kind of creates some, I would say, questions in props mind. So yeah, just mention the referees that are actually willing to offer LOR to you or recommendation letters. And please don't try to fabricate anything. To my experience, there are many students who just write a lot of things on their CV. So somebody, let's take an example. Somebody just have heard of R, or just know 2 or 3 lines of code. And if somebody writes proficiency in R, and like, so if that's the case, and if the students gets accepted, so the professor believes that that student is proficient in R, and he or she doesn't need any kind of help or guidance with that. So after getting into graduate school and having those things in your CV, and at later stage, when you go to prof saying that I need help with these things, that will be a big disaster. So please don't try to do that. So that's all from my side. I hope that was of some use to the attendees. Thank you so much for your patience, and I'm open to any questions if you have. Thank you so much.
Okay. So first of all, so indeed, that was a great session from the researcher. Thank you so much for your presentation and sharing your experience regarding the plant health and your degree. Okay, for the participants, there is some notice. So we'll be having one more presentation session because the time is so we have limited time. So after finishing the presentation session from both speakers, then we'll be addressing the question answer session of from both of the speaker. So without any further ado, let's move to our second language speaker. Our next speaker is Mr. Ramdev Bani, a PhD student in plant pathology with a dual degree in international educational and development at the Pennsylvania State University. So Ram's research interests include the plant health, agriculture policy, and the international agriculture and the development. He is a young scientist member of the World Food Forum hosted by the Food and Agriculture Organization. He actively collaborates with policymakers in global forum to enhance the youth involvement in the agriculture. With over the 7 years of experience in non-profit organization, international agency, academia, and industry, Ram brings a wealth of knowledge to our discussion. Having lived and worked in Thailand, Vietnam, Nepal, and the USA, Ram possesses a unique perspective on understanding agriculture and development in both the global south and north. So, Ram, Dai, the floor is yours now.
Thank you, Upendra Bhai. I'm going to share my slide now. I don't know if you can see. Can you see my screen now?
Yes, brother.
All right, thank you. Well, I'm a PhD student of plant pathology and international agriculture and development at Pennsylvania State University. My research interest is more about integrating the plant pathology, that's a natural science, and social science. So title of my presentation today is Beyond Borders, Beyond Boundaries, an interdisciplinary journey through plant pathology and international agriculture and development. Sorry. I was born and raised in Gorkha. I completed my school from Gorkha, then I moved to Kathmandu for my high school, that's 11 and 12. Then I went to Agriculture and Forestry University in Rampur for my undergraduate degree in agriculture. And during my undergraduate degree, I think Rampur is basically a blank paper. You can write anything on that blank paper. That's the most formative period of my whole career because apart from my study, I was engaged in multiple projects or multiple organizations that helped me to develop other skills. And if you are applying to higher education in America, they don't look into your only grades or GPA or any academic achievements. They generally do a holistic assessment of a candidate, not only your GPA or GRE scores, but your leadership skills. How can you work in a team? How social you are? How well you can communicate? Those are the skills you can develop during your undergraduate study. Because in Rampur, I know there is a lot of politics, but again, you can seek many opportunities. And after undergraduate degree, I went to Thailand in Kasetsart University, where I worked for 6 months on rice blast disease. That was also one of the most important phase of my career, because that was the experience that actually got me interested in plant pathology. And in that research project, I worked on rice blast disease. I was evaluating the genetic diversity of rice blast fungi in Thailand. And then COVID happened. I came back to Nepal. I worked in industry, that's agriculture in Nepal, for almost 1 year. And during that 1 year, I applied to most of the scholarship bi-level across the globe. Let's say Erasmus Mundus scholarship or mixed scholarship, and even in US, I applied to multiple universities, and even in Canada, I applied to multiple universities. And after, along with working in the industry, I was applying to universities and pretty much got offers from multiple scholarship. For example, I got Erasmus Mundus. Even in US, I got multiple offers. And eventually I ended up in Pennsylvania State University because at that point I was aware, or I was able to understand what are my research interests or what I'm going to do for next 5 or 10 years. And one of the reasons why I chose Pennsylvania State University for my PhD is Pennsylvania State University has a unique dual title program that's in plant pathology and international agriculture and development. Plant pathology is more about, I would say, more natural science, whereas international agriculture and development is more social science. And I realized that I want to integrate natural science with the social science. And currently, I'm in my 4th year. I'm trying to wrap up my research and graduate maybe in 1.5 years. Well, my PhD projects, there are 2 components in my PhD project. As I already said, I have interest in plant pathology as well as in international agriculture and development. My plant pathology PhD project is all about identifying the bacteria associated with symptomatic onion bulbs and leaves, and then characterizing their pathogenicity. Here, by the word pathogenicity, I'm saying whether the bacteria can cause disease or not in a qualitative term. There is another word, virulence. Virulence is more about quantifying the ability of any pathogens to cause disease. If you are rating the disease in different scales or assessing the incidence or severity, that's virulence. But if you are just saying whether the bacteria is pathogenic or non-pathogenic, then that's pathogenicity. So why I'm defining these terms here is because plant pathology is more technical, and when you are writing emails to the professors and while you were communicating about your research experience, make sure you are using the appropriate word in your email. And if you use, let's say, if you are doing qualitative assessment of the plant disease, and in the email, if you are saying virulence, then that's the red flag to the professor that you don't have well grasp on the content or on the research you are doing. And copper resistance, and another component of my plant pathology is copper resistance of bacteria. I surveyed onion fields across New York and Pennsylvania throughout 2020 to 2023. And I interacted with many growers during the survey. And majority of the growers I talked with were very concerned about the copper resistance in bacteria. That's because copper-based bactericides is the only, I would say, or primary chemical bactericides available to control the bacterial disease. But if you look into the fungal diseases, there are multiple fungicides that growers can use. Right now, at least based on our experiences in Pennsylvania and New York and across the US, I can say that copper-based bactericides is the primary bactericides that growers are relying on to control their bacterial diseases. That's why then I added the copper resistance component to my PhD thesis, because I believe in consumers, that is growers, demand research priorities. Initially, I was interested more into the molecular, dissecting the molecular biology of another bacterial pathogen, which is Pantheria, that causes center rot of onion. But once I talked with the growers, then I realized that, well, copper resistance is more problematic, and this will add some value to the knowledge we have so far. So that was the component I added later. Here, I'm saying that throughout your PhD, your research project's going to change. So don't get hung up right now in your undergraduate study that, well, I want to do this and that. Because with time, with experiences, your interest will change and you will get a chance to adjust your projects accordingly. But right now, it should be clear that you will be doing the graduate study, let's say in a broader field, plant pathology. And it's good to have more specific ideas, but I'm just sharing that your interest going to change with time and with experiences. And my next component is understanding stakeholder perceptions on the use of wastewater in agriculture through system thinking approach, a case study from Kathmandu, Nepal. And this is my international agriculture and development component, which I am planning to do a fieldwork maybe this summer. And here with the word system thinking approach is like understanding the perceptions of stakeholders. That could be growers, that could be consumers, that could be environmentalists, that could be policymakers, bringing them together and what is their different perceptions. And this system thinking approach is very important if you are working in a very complex system to understand what's going on. And it will help to identify trade-offs and then maximize the efficiency of the system to get to amplify the benefits. I want to give you just a brief overview of my projects because Ritesh sir already covered most of the things you need to know for the graduate school application. And in the question and answer session, you can ask if you have any specific questions for me. Well, in my first project of the PhD, I did bacterial survey for 3 years from across onion fields of Pennsylvania and New York. We sampled 30 fields and we collected more than 500 symptomatic plants or bulbs. And then from those 500 onion samples, we collected or we recovered 1,957 bacterial isolates. And of those 1,957 bacterial isolates, we identified them to the genus level, and we identified around 1,165 bacterial isolates. Well, one of the hypotheses of my first project, there are gonna be huge diversity of bacterial genera or bacteria associated with symptomatic onion bulbs and leaves, but a narrow spectrum of those bacterial genera will be pathogenic. As per my hypothesis, we got 53 bacterial genera. 14 of them were exclusively from New York, 19 of them were exclusively from Pennsylvania, and 13 bacterial genera were present both in New York and Pennsylvania. And of those, just to give you a context, in New York, there is a region with muck swell. Muck swell is a highly fertile swell. And in that muck swell, farmers grow onions, that's a monocropping, year after year after year. And those onions are grown in a very large scale. Whereas in Pennsylvania, onion is cultivated pretty much same manner in Nepal. The growers are smallholders. They have a farm size of around 0.2 hectare to 0.6 hectare. they do mixed cropping, and they do have a crop rotation. So I'm just trying to make a context that you can see some difference in bacteria associated in the symptomatic onions of New York versus Pennsylvania. Here in New York, we found Burkholderia as the most predominant bacterial genera, whereas in Pennsylvania, Panthera is the most predominant bacteria. And this is something we expected because Burkholderia is more soil-borne pathogen. And when there is monocropping and crop is planted year after year after year without crop rotation, it may be that Burkholderia inoculum compiled in the soil for years. Whereas in Pennsylvania, we do have a Panthera, we do have a Burkholderia that's below Panthera. Just a key takeaway of this slide is cropping system influences the bacterial genera present in the field. And after the identification of bacterial genera, we were interested to identify whether those bacteria were pathogenic or non-pathogenic. First, for identifying the pathogenicity, we used 3 assays. First is red skull necrosis assay. Red skull necrosis assay is basically a very resource effective or efficient tool. Here we cut the onion bulb into small pieces, and then we inoculate the bacteria into those small, let's say 4 centimeter scales, and then we incubate it for 4 days. Then if it produces any necrosis, then those bacteria were grouped under pathogenic or positive, and if they did not produce any necrosis after 4 days of incubation, they were grouped as negative or non-pathogenic. And in the red skull necrosis assay, we found that around 75% of the Burkholderia were again pathogenic, whereas around 44% of the Panthera were pathogenic, followed by Pseudomonas aerobacter and other Lichobacter, Ronella, Serratia were just isolated cases with 1 or 2 pathogenic isolates. I already said red skull necrosis is a resource efficient assay just to screen if you have a large number of isolates. So it cannot detect the weak pathogens. Therefore, we selected these non-pathogenic isolates, a subset of this green section from the red skull necrosis or foliar assay and bulb assay, and we again got the similar results here. Let's say around 15 isolates out of 18 isolates that were negative in red skull necrosis were positive in foliar assay. So overall, what I want to say here is that a large percentage of Burkholderia and Panthera were pathogenic, but out of 53 bacterial genera, just few bacterial genera, Burkholderia, Panthera, Pseudomonas, Enterobacter, about 1/5 of the total bacterial genera identified were only pathogenic. Then that was my first project of the PhD. Then I worked into the copper resistance of those bacteria, more specifically Burkholderia strains. And for first was to just screen whether these bacteria were resistant or not. For this, I used a media amended with different concentration of copper sulfate pentahydrate in a concentration range of 100 ppm, 200 ppm, 300 ppm, and 400 ppm. These are the 3 bacterial isolates we isolated from Pennsylvania. And you can see how they have a differential growth pattern at different concentration. This is a control. In the first picture, there is a confluent growth, very well grown. 100 ppm, still there is confluent growth. 200 ppm, still there is there is confluent growth. At 300 ppm in the middle isolates, 22Pa043, you see some non-confluent growth. Non-confluent here is if you have a district colonies and they are very distinct, or they have very distinct boundary, those I have categorized as a non-confluent. And this tells that at 200 ppm, all these three isolates were resistant. And at 300 ppm, first and last isolates were resistant, but the third one is moderately resistant. And at 400 ppm, still you can see some small, distinct, discrete colonies, but the last one, you have a confluent growth. Your confluent growth is basically individual bacterial colonies. Those are densely packed and they have a slimy or smooth surface. And if a bacteria produces confluent growth at a given concentration, then I have categorized them as a resistant or copper resistant bacteria. Whereas if a bacteria is still producing colonies, but those colonies are very discrete with distinct boundaries, those bacterial isolates at the given concentration, I have categorized them as a moderately copper resistant. And if the bacteria does not produce any growth at all, then those bacterial isolates have been categorized as sensitive. Just a preliminary results, I so far completed 33 Burkholderia strains, and out of 33, this is a control. That means all the 38, not 33. Yeah. Out of those 38 Burkholderia strains, all the 38 Burkholderia strains grew in control. That means without copper. And in a media with 100 ppm of copper sulfate pentohydrate, even at 100 ppm, 38 Burkholderia strains produce confluent growth. But as we go to 200 ppm, we see some deviation here. Let's say about 87% of the Burkholderia isolates or strains were still growing in a confluent manner, and those were resistant, whereas 2.6% produced non-confluent growth, and those were categorized as moderately resistant. And I just want to give you a view why this is necessary. Then what? Why do we have to care? Why the growers have to care? The in vitro assay, and these are just in vitro assay, right? It may not correspond to the field results. You need to do this because in the field, resistance is noticeable only when there is a good population or one good enough population of bacterial, like copper resistant bacteria in the field. But if we do this in the in vitro, then it will give us a sense that what's going on in the field, even though it may not 100% correspond with the field, but still it can give us a sense that, well, there might be some resistance, and then we can devote our resources or prioritize our research on this area. And my next project, this is just a screening of the Burkholderia strains, whether they are resistant or non-resistant. After I finish that screening, I'm planning to identify what are the copper-resistant genes present in select strains of Burkholderia. For this, this is the bioinformatic workflow I'll be using. I will be using the whole genome sequencing. After the whole genome sequencing, I will get the raw reads, and the reads are the sequence of fragmented DNA. Once I do the quality control edit and remove the bad sequences, those are called trimmed reads. And those trimmed reads are then used for de novo assembly. You will be using the softwares for this, and this software will then produce contigs or assemblies based on the overlapping. And those assemblies were then those assembly will then be used for annotation and annotate the genes. And so far, based on my understanding, there hasn't been any research, at least in New York and Pennsylvania, based on my understanding, even in USA, on identifying the copper resistant genes of Burkholderia. But there has been a lot of research on Pseudomonas and Xanthomonas. So for the gene mining, I'll be using the homologous genes from Pseudomonas. And for this, I'll be using the different tools and resources like Genius, Blast, Uniprot, and CBI. And after I do the gene mining, then I will also be doing phylogenetic analysis to understand the evolutionary and functional history of those genes. And again, why is it important? Why am I doing this? Because after this what we can do that could be post PhD work. If I get into the academia and if I become a professor, then I will build up my research project on this. My first project will be developing a diagnostic assay to screen copper resistance in Burkholderia. Or my next project could be functional characterization of these copper resistant genes to see if a bacterial strain is exposed to different concentration of copper. How does those copper resistant genes upregulates or downregulates, or how do they respond to the exposure of the copper? So these are the future projects which I'm gonna do after my PhD if I get into the academia. Again, if I go to international organization or non-profit organization or policies, then antimicrobial resistance is a key issue, right? So I can use this knowledge in the policy making as well. And my last project, that is international agriculture and development component, is about understanding the wastewater irrigation. And these are the just outline of the methodologies I'll be using. And I'm planning to come to Nepal in April or May to do the field work. And if anyone is interested, I might be hiring research assistant or research intern for this project. So if you are based in Kathmandu and would be interested in this project, then feel free to contact me. I might be hiring someone in April or May. Understanding the wastewater irrigation. First step is to identify the study area. I'm right now considering Bhaktapur because Bhaktapur has a good, has some pockets of vegetable production regions where they are still using the wastewater irrigation. Then I will identify the growers and select them for the interviews. I will be doing interviews and identification of key variables. I'll be using the causal loop diagramming. I don't know if you are familiar with causal loop diagramming. It is a tool used in system thinking research. And basically, if you have a very complex system with interdependent variables, then this is a very good tool to visualize what's going on in the system. And last is statistical analysis. This is just a causal loop diagram of wastewater irrigation in Kathmandu or in Bhaktapur based on available literature. Here I have identified 5 different variables. Let's say wastewater irrigation. Fertilizer cost is one of the variables that drives wastewater irrigation. And causal loop diagrams has 4 components. One is variable, another is arrows, another is polarity, and another is loop. Here, let's say, if fertilizer cost increases, then growers gonna do more wastewater irrigation. That means both are moving in the same direction, right? If they are moving in the same direction, that is annotated as a plus sign. But let's say when growers do a lot of wastewater irrigation, it's gonna pose more health risks, right? And when there is more health risks, then growers gonna become less positive on the wastewater irrigation. That's why it has a negative sign. Whereas if growers have other clean sources of water, water availability is high, then growers gonna use those water, right? Not the wastewater irrigation. So here I have annotated it as a negative sign. And here are different loops. Here, this is one loop. Wastewater irrigation, if the fertilizer cost increases, wastewater increases. That means changing one variable is driving or amplifying the whole system. That's a wastewater irrigation. That's why this loop has been annotated as a reinforcing loop, whereas this is a balancing loop. When there is high wastewater irrigation, there are gonna be more health risks. And when there is more health risks, growers gonna become less positive on the wastewater irrigation, and there are gonna be less wastewater irrigation. That means this is a balancing loop. It is balancing the system. And if you represent this whole system in a graph, because of this reinforcing loop, the system gonna grow in an exponential manner, but there is still balancing loop that will balance the system. And why this is useful? Because we now visualized a very complex system, and we can identify what are the possible steps of intervention. For example, here in this point, policy making and infrastructure building can come up. Like Department of Health can do some intervention in this step. Similarly, in this step, government can come up with some fertilizer manufacturing or other alternatives or policy making. Even here, if a government invests in different wastewater treatment infrastructures or wastewater irrigation policies, then it can definitely changes the graph. So I can use this knowledge or skills even in plant pathology. Plant pathology is a very complex system. If you go into the field, you will see like very complex system. Fertilizer, irrigation, environment, these are the factors that are influencing the disease development and management. So these are the skills which can be easily transferred to multiple areas. That's why I really like doing the interdisciplinary research and getting out from the silos and getting to integrate the different disciplines and skills. Key takeaways, I did a comprehensive survey to get a snapshot of bacterial pathogens across Pennsylvania and New York onion fields, and our survey revealed that there is a wide spectrum of bacterial genera associated with symptomatic onion bulbs and leaves, but only 1/5 of the bacteria are pathogenic. And my next project is understanding Burkholderia species and their copper resistance. And my last project is understanding wastewater irrigation in vegetable production or onion production in Kathmandu, Nepal by using community participatory and system thinking approach. And beyond academia, which I highly recommend to you or attendees, because it's all about graduate school is all about learning different skills that can be used in your career. For example, I'm doing a research. I can definitely learn skills about designing experiments or ideation first, that is like identifying the key research areas, then hypothesis development, proposal development, then designing the experiment, recording the results, analysis of those data, and then publishing. Those are the key skills. Those are the hardcore skills you will definitely get in the graduate school. But again, if you don't have leadership skills or communication skills, then you're gonna go behind, because this world is highly competitive. And if you don't have those soft skills, then you may lag behind. And in the undergraduate school, basically I'm talking with my own experiences from Rampur. It's a good school to develop those skills, because you have a lot of free time there. You can engage in, if you want to like music, go engage. If you want to learn guitar, go. If you want to sing or learn singing, do that. If you want to engage in engage in different clubs or activism, then do that. It will definitely pay you later in the life. And with that, I'll just end my presentation, and feel free to ask me if you have any specific question. Thank you so much.
Okay, so all right. Indeed, it was a great session. So thank you so much, Ramdev, for letting us know regarding your journey and the PhD project that you are currently working on. So it feels so amazing and motivated to see such progress and research ongoing on the plant pathology aspect. Now we had the privilege of hearing from both of the speakers. Having said that, I would like to open the floor for question and answer. So, dear participants, if you have any kind of questions to our speaker, you may raise your hand, unmute yourself, and ask the specific question to the speaker that you choose to come from the answer. So, if you are not feel free to ask the question from the mic, you can also drop your question in the chat box so that we can address those questions. So, the participants, the floor is yours now. If anyone has a question, then you can raise your hand.
Excuse me, please feel free. I mean, it doesn't matter, who answers. I think we are on the same boat. We'll try to figure it out. So I think most of the attendees, they are all from Nepal. So just for the sake of convenience, you can even ask in Nepali. And I
Yeah,
would
I
also
agree.
in Nepali.
Yeah.
Okay, so still we have, okay, so we saw the response from Aman Mehta. So you can unmute yourself, brother.
Okay. Hello, Ritesh sir. Hello, Ram sir. Warm greetings to both of you. And thank you for so much amazing presentation from both of you. And I do have some queries, and I hope you both will address that. And my first query is with Mr. Ritesh sir. It is, sir, like you already have mentioned about the subject line, like you said, assistantship, and then a proper designation of your specific criteria, like you are interested in phenomics or machine learning, or either on plant pathology or any of the discipline. In recent time, while I have mailed several professors, I used to do progressive. It's like a different kind of subject line. I got to know about from seniors whom I was in a regular contact. So the subject line goes like this: like a prospective MSc student interested in horticultural science, or prospective MSc student interested in agricultural extension, or like that. And I thought that the subject line that you have discussed is quite unique and very concise one. So does it matter to write like a prospective MSc student interested in horticultural science or anything?
Thanks. I think Aman, right?
Yes, sir.
So I think your subject line is perfect. Prospective, master student, it's same thing. They are just synonyms. Um, so in my experience, I always I think profs they're super busy. Like my prof, he gets like 200 emails day from just graduate students, and he doesn't have time to open all of them. So they just go through the subject line, and if you clearly mention what are you interested in, there's a high chance that they will open your email and at least read. So it doesn't matter, but it increases your chances. So I think your subject line is perfect. I think Ram might have better idea on this, so
Yeah, I think you covered pretty much, but I would suggest be concise and direct. And there is cultural thing between Nepal and US or Canada. We Nepalese tend to be overly polite. Prevent that. Just be direct and concise because professors uh, don't have time to go through all the long-winded emails, right? Just be direct. What do you want and what do you have? Just do that in the email.
Okay, thank you so much, sir. And I do have some couple of questions, and if you are free, then I think I should ask. Okay, so my second question is like Mr. Ritesh sir has mentioned about the length of the CV that it should be of around 2 or 3 pages. But after seeing several award-winning scholarship winning the sample of SOP or CV, I have crafted a very good CV. I think I have crafted a good one, and it has got of around 5 to 6 pages, where I have mentioned the key points like the grant, the education, the resource experience, my teaching experience, all the relevant skills related to lab work, related to field work, related to communication, leadership skills, and publications, and my undergraduate experience. So while including all of that, I don't think that I have so much of room to exclude all of them to shorten my CV. And it has got a 5 page. And when discussing with some of my seniors, some of those who have already got assistantship in a foreign country, they do have suggested me to shorten the CV. So is it good for me to shorten to up to 2 3 pages, or should I be concise with 5 or 6 pages? And one more, sir, like for every different professor, for example, I'm mailing you for today, and then for tomorrow, and after some day I'm going to mail Mr. Ramni Upanishad. Then do I have to craft my specific CV, my mail according to the research interest of your individually, like SOP, like mail,
Yes.
CV, everything? Every time do I have to change its context, or so a specific CV will work for the specific arena of those specific research area? Like for both of you are from plant pathology, so do I need to send same specific CV for both of you, or do I need to make a separate one for both of you?
Okay, thanks, Soman. I think you can reduce your CV. Probably you might have mentioned like all of your skills. Like maybe you have mentioned microscopy, and you have elaborated like I did this microscopy, I did this. So you don't have to mention all of the things. Suppose for the sake of example, if somebody has experience with PCR, gel electrophoresis, DNA quantification, they don't need to mention everything. You can just write general skills in molecular biology, because I believe that props, they're very busy. They don't have a lot of time to go all of your like CV. Like let's suppose if you have some interesting things in your CV on the fifth page or fourth page, uh, there's very little probability that would be seen by the prod, because they generally just scan through that. They just look for the keywords. And I think if you can simply there's different referencing styles. So in order to list your publications, you can follow Sitalu, or just you can mention the link to that publication, or you just don't need to mention all of the author's name. You can just write Mehta et al. 2020. What's your title of the paper? And just write the link. So that's how you can reduce it. I just suggested it, but it's not really hard and fast rule that CV must be 3 page, or 4 page or 5 page. So for the purpose of getting some kind of attention from the prof on the first side, I suggest that you keep the CVs concise. And what was your next question?
Do I need to keep my both mail and CV separate one for both of them, professor that I'm going to mail, or
Oh
should
yeah,
I?
definitely, definitely. So professors, they generally like to see your career goal. So suppose you are applying for a plant breeding opening, and if you have written like my career goal is to become a plant pathologist, or my